Good afternoon, everyone. I am pleased to present to you our study entitled Vitellogenin Expression and Synthesis Provide Evidence of Endocrine Disruption in Juvenile Nile Tilapia from the Lakes of San Pablo, Philippines. The National Institute of Environmental Health Sciences, a group composed of public health experts, toxicologists, and environmental scientists, define endocrine disruptors as chemicals that may interfere with the body's endocrine system and produce adverse developmental, reproductive, neurological, and immune effects in both humans and wildlife. In recent years, evidence of surface water contamination with endocrine disruptors has accumulated, which led to its inclusion among emerging contaminants. Since the 1990s, a variety of chemicals have been classified as endocrine disruptors. These include pharmaceuticals such as synthetic hormones. We have industrial additives and byproducts such as bisphenol A, agrochemicals such as cypermethrin and other pesticides, consumer products such as triclosan, and even hormones, natural hormones such as estradiol. In this schematic diagram, the routes of entry of these endocrine disruptors into surface waters is shown. Basically, these compounds enter surface waters via landfill leachates, wastewater, or via surface runoff. As a result, endocrine disruptors occur as complex mixtures in the aquatic environment. Exposure of fish to these compounds has been associated with changes in biochemical, physiological, and morphological characteristics. The smaller and lesser studied 7 crater lakes of San Pablo are likely to be polluted by endocrine disruptors emanating from sewage discharges, intensive aquaculture, and other anthropogenic activities, hence this study. The objectives of this study are to determine endocrine disruption in caged juvenile Nile Tilapia using frequently utilized biomarkers for endocrine disruption in fish. These include Vitellogenin or BTG mRNA expression in the liver, as well as the detection of Vitellogenin or VTG levels in the skin mucus. VTG is a yolk precursor substance typically produced only in mature females during the process of oogenesis. Juvenile fish do not synthesize VTG. Its presence, therefore, in the skin mucus and the expression of the VTG gene transcripts in the liver would indicate environmental exposure of that fish to endocrine disruptors. This is because the liver cells of juveniles can synthesize VTG when stimulated, particularly with exogenous or environmental concentrations of endocrine disruptors or endocrine disrupting substances. The other objective is to confirm the presence of endocrine disruptors in water samples, specifically estradiol or E2, a natural hormone that is excreted together with the urine and feces of vertebrates, and bisphenol A, an estrogenic industrial chemical that has been used in the manufacture of plastics and resins. Both are common contaminants of aquatic ecosystems. For the methodology, the study occurred in 3 phases and was conducted from August to October of 2019. Phase 1 involved setting up of fish cages in identified aquaculture areas of each lake and caging juvenile fish for 30 days in these structures. To minimize the exposure of fish to phytoestrogens in the diet, which could influence the results, they were given natural feeds such as azolla and freshwater shrimp. The location of the fish cages in each lake are shown in the table. A reference group of fish was kept at the UPLB Limnological Research Station and received the same diet. On the other hand, water samples were collected from 3 sampling points per lake within the vicinity of the fish cages and at a distance of about 50 meters from each other. Phase 2 involved the analysis of fish samples. After the caging period, fish were collected and brought to the laboratory, and mucus swabs were obtained from the skin of anesthetized fish. Vitellogenin was quantified from these samples using enzyme-linked immunosorbent assay. Thereafter, fish were euthanized and liver samples were collected. These were subjected to VTG mRNA isolation and detection of VTG genes A, B, and C using RT-PCR. The last phase involved the analysis of lake water samples, wherein samples were collected and placed into BPA and phthalate-free containers with screw cap cover and transported on ice to the laboratory. Each sample was then filtered, subjected to solid phase extraction, and measured for estradiol and BPA using enzyme-linked immunosorbent assay. For the results, detectable levels of vitellogenin were measured in the skin mucus of fish across all lakes, with values ranging from 12 nanograms per ml to 122 nanograms per ml. Higher VTG levels were obtained in palakpakin and in mohicap fish versus the samples obtained from the reference, Yambo, Pandin, and Sampaloc samples. In contrast, pre-exposure vitellogenin values in the skin mucus of laboratory-raised juvenile Nile Tilapia only averaged to less than 10 nanograms per milliliter. All three vitellogenin gene types were expressed across all sites with a percentage range of 10 to 60% for VTGA, 85 to 100% for VTGB, and 55 to 100% for VTGC. VTGB and C were more highly expressed than VTGA. The implications of this result, however, requires more study. VTGA was not even expressed in reference 1 on this particular figure. So reference 1 is composed of juvenile Nile tilapia depurated for 7 days in fish tanks with dechlorinated tap water. Table 1 shows the E2 and BPA levels in all water samples. Higher E2 concentrations were measured in each lake than in the reference site. However, E2 levels in Bunot, Calibato, and Sampaloc lakes exceeded the limit of detection of the assay used. Extrapolation of data showed that the concentrations of E2 in the water samples in these lakes were 1.52 for Bunot Lake, 1.13 micrograms per liter for Calibato Lake, and 1.17 micrograms per liter for Sampaloc Lake. BPA was likewise detected in all lakes, as well as in the reference site. However, most of the BPA levels in the lakes exceeded the limit of detection of the test used, with the exception of Bunot Lake and Sampaloc lakes. From extrapolation, the BPA values range from 13.26 micrograms per liter in Pandin Lake to 19.39 micrograms per liter in Palakpakin Lake. Let's now go to the conclusion. Our findings add the 7 lakes of San Pablo to the list of freshwater systems contaminated with endocrine disruptors. And this is evidenced by the observations and data obtained on the VTG levels in the skin mucus of fish, as well as the expression of the VTG gene in the liver samples of fish. And this was further confirmed by the detectable levels of E2 and BPA that were measured in water samples. Future research should identify specific sources of endocrine disruptors in the lakes, because by identifying these specific sources, it would be easy or much easier um, to set up or to formulate mitigating measures that would minimize the release of these substances into various waterways. Another is to determine the probable impacts of endocrine disruptors on fish populations and investigate the contamination of ED and its effects in other freshwater resources. Although EDs are currently unregulated as environmental pollutants, the Interorganization Program for the Sound Management of Chemicals has recognized this phenomenon as a global threat that needs to be resolved. And this is a perception that has been ascribed to the following observations in the human population: the increasing incidence of reproductive problems and hormone-related cancers, such as testicular cancer and breast cancer, lower human fertility rates, and increasing human exposure to endocrine disruptors, as well as a growing number of chemicals that are screened as endocrine disruptors. Endocrine disruption has also warranted the adoption of precautionary approach in environmental policy in the US and in Europe. In effect, relevant programs have been initiated in these regions to further assess and regulate endocrine disruptors, even without rock-solid scientific evidence, in order to prevent harm to humans and the environment. We would like to thank the DOST Grants and Aid Program for their funding support to this study. Thank you very much for your attention.